Well, first of all, let me thank you guys both for the lovely introduction, and let me also thank all of you who are here for coming, because today is quite a day, okay? So let me start this the way I really wanted it. We're gonna definitely last longer than you're supposed to, but yesterday I was just a buffoon of a candidate for the Republican Party. Today I flew in. I didn't have time to see the hairdresser. That's why this is messy. But today I'm President of the United States. And as President of the United States, whatever happened to my first slide? Wait a minute, what'd you guys do to my slide? I came from the East Coast, and my title, as you can see, is Making Membrane Transport Great Again, okay? So that's to start with. Now, off with this, the end of that, and we can start with a real talk. Okay, so this is my favorite title these days, which is Dr. K. Chase's The Great White Permease, or How I Stopped Worrying and Learned to Love LacY, Permease. I doubt very much whether I will finish this in 45 minutes, so be prepared. I'm going to give this in such a way that in the beginning, I'm going to go very fast over things that you don't have to think about. It's stuff that's very straightforward. And when it's time to think, I'll tell you, okay? So, okay, starting in the beginning, there were membrane vesicles. You already heard that story. They're called Kabakosomes. I never use the term primarily out of modesty. However, these Kabakosomes transport all kinds of stuff. And once we got them to transport, which is shown in these old slides, even that was difficult because you had to find out that there was a D-lactate dehydrogenase in the membranes that was really responsible for energizing them. And at the time, we didn't know what energizing meant. It just gave you lots of transport. And you could also do this with artificial electron donors, like that one out there. Why is this not working? Maybe because I didn't turn it on. That would help. Okay, so as you can see there. All right, so one of the, whoops. Let me go back to where we were. Yeah, okay, so you could also use artificial electron donors like ascorbate and phenazine methosulfate, which bypass the dehydrogenase and work very effectively to drive transport. One of the things that we were able to do with this was to demonstrate that every single membrane vesicle in the preparation transports, every one. And that took forever to do. So here's the way it was done. Very quickly, we had a compound called vinyl glycolate, which you can see up here. It has a double bond there, and it's a lactate analog that gets transported through the membrane inside. The dehydrogenase treats it like lactate, and it gets converted into this double bond compound, which can be attacked in a Michael's addition to that carbon, and you get covalent labeling of the membrane. And it turns out, as you can see from the next slide, that the rate limiting step for covalently labeling membranes is transport. So doing this, what happened was that we could then take one of the nice things about working at a place like the Roche Institute was they have incredible chemical facilities. So my good friend Arnie Liebman, who was head of the isotope synthesis group, there made us very hot radioactive vinyl glycolate, this compound. And we did this particular experiment, and what you see over here are radioautographs of membrane vesicles at the top labeled by transport versus acetic anhydride, which non-specifically labels lysine residues. And if you look at about a zillion membrane vesicles, you can't tell the difference. So every single membrane vesicle transports, okay? Then came the bioenergetics era. Once it was, you got to realize that no one wanted to believe membrane vesicles in the beginning. At first, they didn't want to believe it. Then after it was clear that they worked, they had to be mixed. The orientation had to be this, that, or everything else. And once this was done, showing that every single vesicle transports, you can't very well argue that there are too many that are inside out, right? It was impossible. So then became this era of bioenergetics, and in the bioenergetics part of this, it was initially hypothesized by yours truly that membrane transport proteins were components of the respiratory chain undergoing sulfhydryl disulfide interconversion, which accounted for transport. And I won't tell you what the evidence for this was, but it was good enough to make Leninger's textbook in 1972. Originally, it was known as the Kaback model for transport. As it began to fall into disrepute, it became known as the Kaback and Barnes model, my postdoc. As it fell into further disrepute, it became the Barnes and Kaback. And now it's known as the Barnes model, okay? Because it ain't true, okay? So at the time, what really was true, or turned out to be true, was a hypothesis at the time, which is really now should be called a law, called chemiosmosis, which was dreamed up by this iconoclastic Englishman named Peter Mitchell, who came up with the idea that the basic energy trapping devices of living cells, respiration or light, give rise to an electrochemical proton gradient, which consists of a membrane potential, an electrical potential, and a gradient of pH. And that this is the driving force for all kinds of things like oxidative phosphorylation, active transport, transhydrogenase, the bacterial flagellar motor, and lots of other things. Okay, and it turned out that once it became clear in my own lab, when we were able to measure pH gradients and membrane potentials in microscopic systems quantitatively, I might add, we were able to demonstrate that Mitchell's chemiosmotic hypothesis was correct for all kinds of things. And that's what are shown here, but I'm not going to tell you about that. The idea was to focus upon one of these proteins and try and, you could either, well, let me put it this way. You could either take the techniques that we developed to measure membrane potentials and pH gradients, go into intracellular organelles, whatever you like, and make those measurements. Or if you're crazy, you try to figure out how a transporter actually works, okay? And as you already heard from both introducers, I am a little bit crazy. So I decided to go after a transport protein. The transport protein to go after at the time, and probably still is the lactose permease from E. coli. As you already heard, at the time that I was messing around with this, when I was in my younger days, there were still arguments about whether there were membranes, let alone proteins that transported things through them. And the only protein the only transporter for which there was evidence that there was a protein involved was the second gene encoded by the lac operon, the LacY gene, which encodes lactose transport. And that immediately implied that there had to be a protein involved, okay? So that was the one to focus on, and that's the one I focused on ever since. So it's been a long love affair with this crazy proteins. Okay, so then this began the chase for the great white permease. And initially, we started with it because in the late 1970s, it became the first gene encoding a membrane protein to be cloned and sequenced by Benno Mullerhill in Cologne, Germany. And we began doing molecular biology in the form of site-directed mutagenesis, which I thought was the greatest technique since Pepsi-Cola. I thought this was absolutely spectacular. I used to tell people this is spectacular. It's like playing God, but of course you're playing God with a blindfold if you don't have a structure, which we didn't, and nor did anybody else. But we started doing site-directed mutagenesis, which led to cysteine scanning mutagenesis, and But what happened was this. Ultimate fears were realized, as you can see. And if you can't see, you're blind, okay? You understand what this shows. Yes, I hope. Okay. Anyhow, so now between 1983 and 2003, which was the year in which we finally ended up with the structure, a lot of things happened. So I'm going to go through these very quickly. Each one of these little blurbs deserves an entire seminar, but I'm not going to talk about them. So functionally and structurally, the thing is a monomer with one sugar bound per mole of protein. From hydropathy plots, we guessed that there were 12 transmembrane helices. Turned out to be a lucky guess, because it was true. It turned out to be true. The protein is conformationally extremely dynamic, and I'm not going to tell you much about that. You'll see a lot of that. It was 85% helix by circular dichroism, which turns out to be the best method for assaying helicity. Okay, the other techniques are nowhere near as accurate as CD. Cysteine scanning mutagenesis, that I already mentioned, was developed as a result of all of this. There were 9 irreplaceable residues out of 417. What I mean by that is the only way. Let me go back. Sorry, I didn't mean to do that. This is very sensitive. So the only way you can actually figure out what residues are absolutely essential for your catalysis of whatever you're measuring is to actually mutate every single residue. You can't do it by standard genetic techniques. It just won't work, okay? So it turned out that out of 417 residues, there are 9 residues in this protein that are really complete knockouts, and you can't replace them with anything except in some cases the most conservative replacement possible. Okay, what happened as a result of all of this was we had this library. The idea of making cysteine scanning was not, as people thought, just busy work, making everything mutate into a cysteine. The idea at the time, having had already tried to make crystals out of LacY for a long time and failing miserably, I had decided that if we made a whole library of cysteine mutants in a gene that was engineered to have a unique restriction site every 100 base pairs, we would be able to get, by cross-linking, by 5-hole cross-linking, in easy ways, you'd be able to get helix packing, okay, structure. And this was actually published, a paper like this, as a result of lots of cross-linking. And as you will see, you could do cross-linking till hell freezes over, and you'll never get the structure, okay? No way, as you will see in a second. So it led to a model for helix packing, and the real summation of this period of time was a really beautiful experiment carried out by Kazanobu Matsushita when he was in my lab, in which he took purified LacY and purified cytochrome O, which is one of the terminal oxidases in E. coli. And with those two proteins reconstituted into proteoliposome, he could reconstitute active transport, which was really beautiful. Very satisfying experiment. Finally, a structure, the modern era, 2003. So what happened is, here's what the lattice looks like, and you can already tell this is one hell of a lot of helices, okay? And the way this happened, and I'm giving you the short version, is Irina Smirnova and Vladimir Kasho, who were husband and wife and seated in the audience, and whose pictures you're going to see a lot of. And at the time, Joe Werner, who was the technician in the lab, prepared lots of Permease. And every couple of weeks or so, we would send a package to Imperial College in London, where Jeff Abramson, who had said, ho, 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 this thing should crystallize, and we all laughed at him, he crystallized it, and then he and Zoe Wada solved the structure. And Jeff, as you know, is here in the physiology department now. Okay, so here's what the structure looked like, and immediately there was a surprise. Who would figure that a transport protein would be closed on the outside? This is the periplasmic side, that's the outside. And don't be confused by other people's pictures of up and down. It became fashionable for the eukaryotic membrane people to draw these the other way around, okay? And we used to say that was the difference between prokaryotic and eukaryotic membrane proteins. It's become actually a confusing pain in the neck, really. Okay, so we'll get back to here. So you can see it's got two bundles, two 6-helix bundles of mostly irregular helices. The initial crystallization was done with a mutant, C154G. That was the second native cysteine to make a cissless permease. And what it says here is sometimes it pays to be stupid. And this is true. Sometimes it does pay to be stupid. What this means in this case is that at this time we were taking the native cysteines in LacY and changing them to serine, which is okay, or glycine, which is stupid, especially if you're making these substitutions in the middle of a transmembrane helix, because glycine is a helix breaker. However, this particular mutation left us with a protein that bound ligand but didn't transport. And it stayed in the freezer for 10 years because we were chasing the wild type until Jeff finally, on a visit, said this one should crystallize, and it's the one that did crystallize. So, but it was a mutant, and in a second I'm going to show you that it wasn't a monster. The point of this was that just to get it going is that the reason for the mutation is that it's right here, that cysteine, and when you look closely, when you finally get structures, that cysteine that mutated to a glycine is in helix 5, and in helix 1, which is right next to helix 5, there's a glycine at position 24. Now, as initially postulated by Don Engelman and his group, if you have two helices next to each other and at the same level glycines, that allows the helices to pack tighter, because glycine's no side chain, right? And that's what this apparently does, and we have lots of evidence for that I'm not going to show you because I don't have time. Okay, so we're back to here again. Okay, this is the work of Lan Guan in the lab, who finally crystallized the wild type by making sure that it didn't lose all of its phospholipid. And as you can see, the wild type superimposes on the C154G mutant. So the mutant wasn't a monster, clearly. And here's a picture of Lan in her Hollywood mode with wearing sunglasses. She's now at Texas Tech University with her own lab. Okay. It took 10 years to get a different conformation. This thing loves to be in that outward closed, inward open conformation, and it's a pain in the neck because we kept getting the same structure over and over and over again. However, a different conformation came about in the fall of 2013, and it started in the following way. This is the characters involved. It was a collaboration with Bob Stroud's lab, and Hemant Kumar was the postdoc who did the work, actually. So what happened, really, the way this started was, again, Vladimir and Irina did something very clever. What they did was a blast search, and they noticed something very interesting. They noticed that in many of the members of the major facilitator superfamily, of which LacY is the poster child, there are pairs of glycine residues at the ends of helices. You'll remember I told you that glycines let helices pack tighter. So apparently these glycines seal the periphery of the protein. So what they did was they got this bright idea. Let's stick a big bulky tryptophan residue in place of one of the glycines in each pair. And what they got was a protein that didn't do any transport at all, but bound ligand like crazy, okay? really bound beautifully. And this was supposed to give us the open outward conformation, which we never got, but there was then a surprise, there was a surprise. And the surprise was that the thing was, you can see it best in this angle, there was we got a protein that for the first time had sugar bound, and here's the tryptophanes, and the periplasmic side was open, partially open, but only partially. And in order to get from the original inward open, outward closed conformation, and this conformation, you have to rotate around 30 degrees around the middle of the protein, for what that's worth. Although we don't think that it's rigid body movements for sure. Okay, to give you a better idea of what this thing really looks like, is if you do space filling models, what you see is, if you look at the periplasmic side, you can hardly see the sugar buried in there. Obviously, this is too narrow for the sugar to get out of, you think. We may modify this a bit by the end, as you'll see. A slab tells you that the pathway is, in fact, way too narrow for the sugar to get out, and the cytoplasmic side is closed. What this tells you is that there must be, I'm going back again, that there must be an occluded intermediate involved in the transport cycle. Was this? They're clearly. What happens here is the sugar gets bound. Probably it's got to be more open than what you look at, than here. It has to be more open, obviously. How more open, we're not sure, as you'll see. But the sugar gets in, then it closes around it, supposedly, to make this occluded state. And then it's that state that's the high energy intermediate, which is what I'm going to tell you about most of the time. So the sugar gives enough density that you can really get the interactions between the side chains, which we knew about. We knew which side chains were involved in binding, but we didn't know which part of the sugar they were bound to. Now, all the specificity of the protein is in the galactosyle end of the molecule. So all the specificity is down here. Galactose itself has terrible affinity. But if you stick garbage on the one position, as shown here is the glucose molecule, or this is TDG actually, it's another galactose. But if you stick glucose and you make lactose, or you stick particularly hydrophobic stuff, like nitrophenol shown here, you can increase affinity like 1,000-fold, okay? But all the specificity is in this part of the protein, and probably the most important residues are glutamate 269, okay, and this asparagine 272, because it's the 3 and the 4 position that are most important for specificity. Glucosides and glucose don't bind at all. And the only difference between galactose and glucose is that the 4th hydroxyl in galactose points up, and glucose, it's axial, okay? We won't go anymore, we don't need to do any more with that. Okay, so the whole business with the active site looks like this. So the green is the van der Waals surface, and you can see all the gold residues are the ones that are involved in binding. Okay, here's TDG is the symmetrical substrate, the only hydrophilic one that has good affinity. There are a couple of residues that are interesting, like cysteine 148 is famous. That's the original cysteine that Fox and Kennedy labeled protectively in 1965 to identify LacY as a membrane protein. That's protected against alkylation by substrate, and very useful for that reason. This alanine up here at 122, if you change it to a cysteine and put something bulky on it, or change it to a tryptophan, then you make the permease specific for galactose transport, because the second sugar can't fit. It's sterically blocked, which is cute. It's a nice experiment. Okay. One of the things that was, this is one of the things that was interesting was done with a compound called MTS, methane thiosulfonyl galactoside, made for us by Tamás Kalai and Kalman Hidig at the University of Pest in Hungary. This stuff is a suicide substrate for the permease. And what it does, something is happening, the pointer is not working anymore. Did we run out of gas? No, it seems to be. I think it's run out of gas. Is there another pointer? Pardon? What's this? This is something. Here's red. Let's try red. Yeah, this works. Okay, so this stuff, if you make that alanine into a cysteine, this stuff becomes a suicide substrate, sticks there, and now I have to change slides. Let's do it this way. I think this will. Yeah, okay. So this is Lon Guan's work. She showed it's a suicide substrate, and then along came Jeff and his postdoc, Vince Chaptal, who's now back in France, and they got a structure, and the structure, to our great disappointment, was the same old outside closed, inside open. And what that meant at the time, we didn't know, because this was before we had the double tryptophan structure. So what it really says is that in order for the protein to go into the occluded conformation, you have to have a perfect fit between the galactose and the binding site. It can't do it here because it's stuck by this tail, okay? Okay. So the galactoside has to be completely liganded for the protein to go into its occluded state, which means that it's probably an induced fit phenomenon, probably, okay? All right, so there's something called alternating access, which means that the protein can access the binding sites for protons and sugar to either side of the membrane alternatively, just like is shown here. And that was postulated by Peter Mitchell, of all people, in 1968. Although other people are credited for it, I think Mitchell deserves most credit because he's the one that decided that the protein really moves around the substrate. It's not that the protein moves the substrate through it. It just moves around the substrate. Okay, so, whoops, sorry. We have many, many different lines of evidence to support alternating access. Most of the time in this business, people look at a structure, and if it's open on one side, they call it must be alternating access. We have really spent a lot of time with this, with Irene and Vladimir in particular. So I'm not going to give you all the evidence. I'm just going to take two of the ones I like the most, which are fluorescence and more recent stuff with nanobodies, which I'll tell you about in some detail. So first of all, one of the nicest ways to show alternating access was to put a tryptophan on one side and initially a histidine on the other side, because a protonated imidazole quenches tryptophan fluorescence. You can also do it, and it's better to do with a cysteine and a tryptophan, and then put bimane, which is a fluorophore, on the cysteine, and it gets quenched by tryptophan. So anyhow, you put the pairs either on the cytoplasmic side or the periplasmic side. These are two different constructs, of course. It's not the same construct. If you go to the periplasmic construct, what you get is beautiful. Very little fluorescence, you add substrate, and zoom, you dequench because these come apart, right? It opens up. Yes? This titrates with a pK of around 8, which is perfect for a a histidine sitting in the hydrophobic environment of a tryptophan. If you go to the other side, you get exactly the opposite. You get quenching as these come together. So when the outside opens, the inside closes, and it's reciprocal, right? And the rates are approximately the same. So that's nice evidence for that. That's Irina's work. More work from Irina and Vladimir was the demonstration that nitrophenyl galactoside is in perfect position, the nitrophenyl group, to do fret with the tryptophan in the active site of the protein. And this is really cute because you add, the way you do it is you add nitrophenyl galactoside, which has an inner filter effect, so you get a lot of quenching of light. And then you add a sugar like thiodigalactoside that's not fluorescent. You kick off the NPG, and you get an increase in fluorescence, which is due to fret. Okay, so the difference is fret. Okay, a really neat way, and we use all of these things routinely these days. And one of the more surprising things to come out of this, if I can get this straight, is that here's a study where Irina and Vladimir did a study where you take the protein, LacY, either in detergent, in dodecyl maltoside, or reconstituted into proteoliposomes. And our reconstitution system reconstitutes with the protein all in the same orientation as in the native membrane, okay? So what happens in detergent is you get a linear dependence of the rate of binding of nitrophenyl galactoside on the concentration of the sugar, okay? But if you put the protein into proteoliposomes, there's no dependence. It stays at around 20 to 30 per second. What this says is that there's a step before binding that's important, and it's to open, in other words. In detergent, of course, it's open, because you have an outside binding site and an inside binding site, one of which is open. We don't know which one it is at this stage, but it's not important. The important thing is that in proteoliposomes, there's a step that's independent and probably depends on opening the cavity. And what's interesting is that this number I gave you of 20 to 30 per second is about the same rate as transport is. So it is not unlikely that the rate limiting step for transport is opening the outside, okay? All of this comes together at one point. There's Vladimir and Irene again. All right, so now comes the thinking part, okay? For those of you who are still awake, okay? Yeah, I'm going to talk about mechanism, and because that's the reason for doing all this stuff, okay? So to present all kinds of structure stuff without going into mechanism, to me, is very boring. But so we're going to talk about mechanism, and just to remind you, this is the way the thing works. So if you have an electrochemical proton gradient, the protons go down the electrochemical gradient. The free energy that's released is used by the protein to drive lactose up a concentration gradient, okay? It's always coupled. You never get movement of the sugar without protons or movement of protons without sugar, okay? So if you take, in this case, sugar concentration gradients and push the sugar either in or out, protons will go uphill in either direction, and you get a generation of a delta mu H that's consistent with which way you push the protons, okay? That's very important. So just remember, always coupled. Okay, there's one reaction that I haven't told you about, and that's, there were a couple of them. It's called, they're both the same, actually. Equilibrium exchange and counterflow. What that means is that for equilibrium exchange is you take vesicles or proteoliposomes, load them with radioactive lactose, squirt them into a medium containing an equal concentration of unlabeled lactose, and look at the rate at which you get exchange, okay? The reverse of that is called counterflow, where you take high lactose concentration cold inside, dilute into low concentrations of hot stuff, and you get an apparent initial concentration of the hot stuff because of exchange. You'll see this stuff in a second. The important point is that in these reactions, the proton never comes off. It just sits there. This thing you can take up to pH 11 and it keeps going, okay? So these reactions are independent of the proton electrochemical gradient, as you're going to see. So this is counterflow, the one I told you about. So you have high lactose inside, you get initially 1-to-1 exchange for the hot stuff outside, so you get very rapid uptake, and then when all the inside is exchanged, it stops. And then everything goes back down to the baseline because the driving force is the concentration gradient of the sugar, okay? And it's dependent on pH. The down part is dependent on pH because efflux is dependent on pH, okay? If you do, well, let me go back. If you do this in the presence of a protonophore, come on, baby, like CCCP, which is the open circles, I think it's the open circles. Yeah, the open circles, nothing happens. So this is completely independent of delta mu H, okay? In the case of efflux, what's happening here? Back, come on. In the case of efflux, what happens is if you impose membrane potentials, you do this by making a potassium gradient where you have high potassium inside and you dilute into low potassium outside and you add valinomycin, an ionophore that's specific for potassium and makes the flux of potassium very high. So you make a membrane potential that's interior negative, okay? If that's complicated, I can explain it later for you. Anyhow, when you do this, if you make a membrane potential and you look at efflux, which goes with a proton now, it's a coupled now, right? You can completely inhibit almost with a potential of about 150 millivolts, okay? If you do that with exchange, this one, not a damn thing happens, nothing, okay? So what that means, see, these exchange reactions are in fact alternating access. That's the protein flipping back and forth, okay? And what it means is that reaction is independent of delta mu H, right? This is really important mechanistically because the Mitchell hypothesis, chemiosmosis, is a thermodynamic hypothesis, which has nothing to do with mechanism, okay? So here you start to dissect the mechanism, and it's very important to realize that delta mu H is not involved with the global conformational change that's alternating access, okay? We're going to get to more in a minute. Now we get to some mutants. I love mutants, and these were amazing. We took all of the irreplaceable residues and they're all dead in terms of active transport. But we did efflux, exchange, and counterflow, what I've told you about already. So if you do efflux, all the glutamate 325, these red ones they're all the same really. This is experimental error probably, okay? This is a log plot, okay? The wild type's they're much slower than the wild type, right? But that's a reaction that involves a proton movement. It's efflux, right? If you do exchange, equilibrium exchange, something really dramatic happens. The red ones, these red ones, and the rest of the ones which I'm not telling you, they don't do anything. They do very little. But the arginine 302 replaced with either serine or alanine, or glutamate 325 replaced with any neutral residue, does equilibrium exchange as well as the wild type. And if you do counterflow experiments, you see something really dramatic. The yellow, the green first is the wild type, so you get boom, quick exchange, and then efflux, and this is at high pH, so it's fast. But the mutant, the 325 mutant, glutamate 325, does the in part, but it can't do efflux, so it stays there, okay? Really interesting. What that says is that glutamate 325 must have something to do with protons. Must, okay? and I'm going to tell you what it has to do with protons. There's Nancy Carrasco and Miklos Shahin-Tot, and who did these experiments. Irene Putner's picture I don't have. These are old experiments. Now, one of the things you always read about, in textbooks, is that there has to be, for active transport, there must be a change in affinity on one side of the membrane, right? I chased that for about 40 years, and we could never find a change in affinity, although we thought we had it on occasion. And it turns out that in an experiment that I'm going to show you in a second, that binding, the KD, is the same on both sides of the membrane. And the way that experiment was done was to take right side out membrane vesicles, okay, and inside out membrane vesicles, using that cysteine at 148, where you can measure KDs for sugar by looking at protection against alkylation. And what we did was to do that, and then in right side out vesicles, make delta mu H, but very low delta mu H, and with inside out vesicles, delta mu H, and look at the effect. In right side out vesicles, you would accumulate the substrate if that was what was going on. In the inside out vesicles, the substrate would get pushed out because it's the opposite delta mu H, right? Well, the KD is identical in both cases, with and without delta mu H. So there's no change in KD with delta mu H at all. So what it means is that active transport does not involve a change in affinity on either side of the membrane. So what's happening? We still don't know. I'm going to tell you. Okay, you may have noticed that this is an ordered mechanism, that protons go on first, and then the sugar goes on, the sugar comes off, and then the protons come off, okay? Let me show you that it's really an ordered mechanism, okay? So here's an experiment that was done by Irina that is really a beauty of an experiment. What she looked at was the KD, the binding constant for different galactosides, as a function of pH. And what you see is really dramatic. You see very nice, very good affinity up to about pH 9, 9.5. And then if you start to kill affinity and you can up to pH 11, you have to stop at 11 because it becomes unstable at that point. The amazing thing is that A means that at physiologic pH, this protein really wants to be protonated, okay? Badly, okay? Very badly. What's really incredible, though, is if you take glutamate 325 with a neutral substitution, it's not just alanine, but neutral substitutions, you completely remove the titration. You get high affinity binding up till the time you kill the protein, okay? That says something very important. It says that in order to bind substrate, you can't have a negative charge on glutamate 325. Conversely, in order to bind substrate, you have to protonate glutamate 325. That's coupling, folks. This complicated business that people have been throwing around for years about coupling, this is the most simple example you can think of. It's just protonation of this one acidic group, probably. There's Irina. Now, I told you it looks like there's a PK, right, of 10.5. Well, here's an experiment that was done very recently in France in Petra Hellwig's lab by her graduate student, Natalia I'm not sure I'm pronouncing her name right, Gristic. Okay. Now what she's done is to use a technique. It's basically Fourier transform infrared spectroscopy, which you can pick up, if you're really lucky, carboxylates. And in this case, you can get lucky because this carboxylate, presumably, if we're right, has a PK of 10.5. The rest of the carboxylates, you shouldn't see at all if you go up to high pH. And what you see, if you look at this, first of all, is that there's this, the typical absorption of the carboxylate is at 1743 wave numbers. And this is the signal you get. You get this little blip. But let me just show you that the blip, if you make glutamate 325 into an alanine, that blip completely disappears. So that's glutamate 325 we're looking at, right? And if you look at it as a function of pH, you can see that as you decrease pH rather, the blip disappears. And if it's plotted, you get this. And it's independent of whether substrate is there or not, okay? This has a pK of 10.5. Now this is, I think, is really incredible, because I would have sworn that having worked with this protein for as long as I worked with it, that when I was sure this was going to turn out to be a very alkaline pK, that it would turn out to be like 5, you know? Just because the protein likes to screw you. It has a mind of its own. So this I'm really excited about. Now here's the kinetic mechanism for the way it works, and I think it's pretty much has to work this way. So you bind the proton first, because the pK is 10.5, glutamate 325, then you can bind sugar, then you go to an occluded intermediate, which can open to either side. If it opens to the inside, sugar comes off first, protons come off second, and the unloaded permease goes back down to here, okay? The shaded part is exchange and counterflow, okay? And the fact that the shaded part is exchange and counterflow is proof that this has to be the ordered mechanism. It can't be anything else. And if you sit and think about it, you'll realize that that's true. You cannot come up with another model that explains the way exchange and counterflow works other than this. Okay, so here's how I think this overall, how this thing works really, with a little bit more evidence. So first of all, symport, which is what this is called, in the absence or presence of the proton electrochemical gradient, is the same reaction, okay? They both go with the proton. In the absence of delta mu-H, there's a primary deuterium isotope effect. Now, in enzymology, if you have a reaction that involves protons, you can frequently, if you're lucky, if it's a rate-limiting step, the proton transfer, you can get a deuterium isotope effect, because deuterium makes the 0-point energy of deuterium with its adduct is more energetic than a proton. Okay, so what you can see here is that if you look at efflux, there's a 4 to 5-fold inhibition in the presence of deuterium, with no effect at all on exchange. Okay, none. You wouldn't expect one because there's no proton movement. With efflux, there is. This is another way of doing it directly with electrophysiology, using something called solid supported membrane electrodes, and you get the same inhibition with D2O. Okay, so there's no delta mu-H effect unless there's proton movement. The sugar binding and dissociation, not delta mu-H, are the driving force for alternating access. LacY, let me just show you that even with active uptake, here we're doing potassium diffusion potential driven uptake. So you make potassium diffusion potential with valinomycin, you make a membrane potential that drives transport, and as you can see, there's no effect on the rate by deuterium, and deuterium, in fact, it protects a bit, gives you better results because deuterium inhibits efflux, and you're beginning to run out of steam at this point where you start getting efflux. Okay. All right. We're down to 3, I guess. Yeah, LacY must be protonated in order to bind sugar. Sugar binding involves induced fit, as I told you. Sugar dissociates, releasing the energy of binding, and there's a conformational change that brings arginine 302, which I didn't mention very much, and near glutamate 325 to cause deprotonation. Our biggest problem at the moment is what's the pK when you lose the proton, not when you get it, when you lose it. Because you obviously have to lose the proton for the thing to work. It's got to cycle. Okay, so, but the important thing is that transport is driven chemiosmotically, but delta mu H, I forgot my changer, so I'm messing everything up, sorry. Go up, okay. Transport is driven chemiosmotically, but delta mu H function kinetically by changing the rate limiting step, okay? Okay, so the point of this, of all of this stuff, is that LacY works like an enzyme, but the transition state is the conformation of the protein, the occluded state, rather than the conformation of the substrate. And I think that's probably true for most transport proteins, although they haven't been studied like this. So what's new, you might ask? What's new, and I've already gone past 1 o'clock, but what's new is the use of nanobodies. Nanobodies, if you don't know what they are they're essentially FV fragments, which are made by camels, sharks, and alpacas, okay? And there's a group in Belgium headed by Jan Steyer, with Els Pardon doing most of the work, in which they do an incredible job. We made nanobodies rather, against the double trip mutant. So we get periplasmic open. That's what we wanted, and that's actually what we got. So what you get with this is amazing stuff. So what's new? You get really amazing stuff. These nanobodies they stabilize outward open conformations. And this is what you're looking at here are all binding of nitrophenylgalactoside using the techniques that I told you about, the spectroscopic techniques. You can see that the rates of binding are all linear, so you can get K off very nicely and K on. And what the nanobodies do is they give you a big increase with the wild type with very little effect on the mutant, okay? Because the mutant's already wide open. So what they're doing is stabilizing the open conformation of the wild type. And if you put them in proteoliposomes, it's even more dramatic. You get a really gigantic change. Now, this experiment is one of the beauties. This was done with more than what you're looking at, but what it shows is that if you put if you look at opening and closing, that on the periplasmic side you get dequenching, and different nanobodies behave differently. That's the point. Okay, so it's very unlikely that we're looking at rigid body movements. Okay, what we're probably looking at is a global population of open outward conformations, but they're all slightly different. And it's the same with the inside. The inside you get quenching is the only difference. Okay. So we finally, in collaboration with Ning Yan's group, specifically Xin Zhang in Beijing, have gotten the first nanobody LacY complexes, and this is, you're looking at one right here. This nanobody, probably all of them they all bind to the periplasmic side. That we know. But here you can see that they bind primarily to the C-terminal bundle. And in this case, one of the CDRs of the nanobody projects into the cavity between the two bundles in such a way as to stabilize the opening, okay? And you can see that here. Now, what's neat about all this is this movie which Vladimir made, which you can see. It's worth running it so you can see it, because it really brings up this issue. We put sugar in. These are without sugar, but the sugar's put in here just so you can see where it is. And you can see that the opening is really small. And this particular mutant binds at a diffusion limited rate. So that's all the opening you need to get diffusion limited rates. And it ain't much. So where the biochemistry experiments all say we get this gigantic opening, but and they may do that, but they don't have to work, okay? Now, the important thing is that for these experiments, the real hero is this llama who made the nanobodies. His name is Gamma, and it brings me to another piece of comedy, which is instead of a chicken in every pot, my motto is a llama for every laboratory. Okay? And then I can't resist some of these. So here's the end of this, and you can see what happens. Those of you who have not read Moby Dick should, because this is what it's all about, okay? This is me graduating from Haverford College, and that's me now after 40 years of working on LacY Permease. The great white shark, that's LacY Permease. And that's me, it's really Gregory Peck with my head, okay? Trying to kill the white whale, okay? Now this is some of the people that worked on this project. I just couldn't show pictures of everybody, which I like to do. And as you can see, it's quite an assortment of characters, and I've had a hell of a good time working with all of them, okay? And in particular, two of the best colleagues you can ever find are Irina Smirnova and Vladimir Kasho, who have done just a huge amount of really elegant stuff. And I thank them publicly for that. And there I am again. Okay. So thank you very much.